This is the sixth series of aldehyde ketone lecture, and we're going to be looking at nitrogen nucleophile now. And the nitrogen nucleophile produces two different type of products. One is called imine, and the other one is called enamine. And that has everything to do with what kind of nitrogen you react the aldehyde or ketone with. So since you have not had amines yet in 114, I want to just briefly go over what amines are and classify them so that we could use them in figuring out whether an imine or enamine forms. All right. So so basically, the reaction that we're looking at is the carbonyl plus some kind of amine. And this gives either something called imine or enamine, depending on the type of amine that you react with. And this, once again, is a reversible reaction. You need an acid catalyst to make this happen. And then when you form the product, you also form water as the byproduct. So this is the overall reaction. And like I said, what kind of amine produces imine or enamine? That is based on the amine. In other words, if I have something, amines are anything with the nitrogen attached to an R group. So if you have this, where you just have one R group attached to the amine, this is a primary amine. Whereas if I have two R groups attached, this would be a secondary amine. And then if I have a nitrogen with the most number of R groups, which would be 3, this would be a tertiary amine. And if you go even further and substitute it with the lone pair and the nitrogen, and you have 4 groups on the nitrogen would have a plus charge on it, right? And that is a quaternary ammonium salt, all right? So obviously, in order for nitrogen in the amine to act as a nucleophile and attack the electrophilic carbon of the carbonyl, the nitrogen has to have a lone pair on the nitrogen to be able to attack. So obviously the primary and the secondary with the lone pair is able to do that, and even tertiary in a sense. But quaternary cannot be a nucleophile, right? So that should be obvious at this point. So let's look at what happens when we react with the carbonyl and primary amine. And like I said, this is a reversible reaction. And what happens with the amine is that, primary amine is that, see how you have 2 hydrogens that is available to leave with the oxygen? So what happens in this case with primary amines, you form a C double bonded to N, which still has R group attached to the nitrogen, and what is extruded is the 2 hydrogen and an oxygen, which forms water. All right, this C double bond N is called imine, and this is a pretty simple reaction product for you to predict, because all you're doing is getting rid of the oxygen. Think of oxygen and H2 leaving as water, and in its place the nitrogen is taking its place, and it's going to be double bonded, and R is going to be still attached to that. All right. Now, if you look at the secondary, amine, same condition. you no longer have that luxury of being able to form water, because this nitrogen now only has one hydrogen, not two. So what has to happen is that carbonyl, not the carbonyl, but the carbon next to it, either one, left or right, must have a hydrogen to provide to form the water. So you have the oxygen, one hydrogen from next door, and then this hydrogen forms the water. All right, and we have to go over the mechanism later, but this is just in keeping track of the atoms. All right, so then what happens is when this gets removed, you're going to form a double bond to here and here, and then instead of a double bonded N, you're going to have a single bonded to N. So this is removed, and then you have a nitrogen with two R groups still attached to it, and this product is called enamine. Alright, so the requirement for enamine to form is that you have to have hydrogen on carbon next to the carbonyl. Otherwise, you can't form the acetyl bond C. Therefore, then you can't form enamine. All right. So, if then you look at what would happen if we had a tertiary amine. Now, this nitrogen doesn't even have a hydrogen to remove, and once it attacks, it's going to have to form this quaternary ammonium salt. So when you react tertiary amine, okay, obviously there's going to be no reaction occurring. All right, so you have to keep that in mind, that the primary amine forms the imine, the secondary amine forms the enamine, and the tertiary amine reacting with aldehyde or ketone gives no reaction at all. Okay, so at this point, your goal is to be able to differentiate between imine and enamine, imine versus enamine, and like for example here, you know, which one's imine, which one's enamine, and which one is neither. So obviously, if you look at it carefully, this one, it's got the acetyl bond N with an R group, so that's an imine. This doesn't have acetyl bond N or acetyl bond C next to it, so that's neither. And this one has acetyl bond C with a single bonded N, so that's with a 2 R group, so that's an enamine. All right. You should be able to answer this question, draw these aldehydes or ketones, figure out whether you have a primary or secondary or tertiary amine, and give product if possible. If not possible, then just write no reaction. All right. And once again, we're going to go over this later, but this is a reversible reaction. So an enamine or imine, if you react with water, lots of water, then the reversible reaction will be favored because you're pushing the equilibrium to the left, then you'll get the carbonyl and the amine back. All right. So if you see this problem here, give the hydrolysis product of the following with an acid catalyst. That means you're reacting this imine and enamine with H2O, loss of H2O in presence of acid. All right. Now, why is this chemistry important? Why are we studying imines and enamines? Well, one reason is that a lot of the chemistry that happens in our body occurs via using imine chemistry. All right, so in your book, there is a chemistry of vision on page 935. I've written down here, incline. So I'm going to go over that. I didn't copy and paste it in here because the structures are huge, and you could take a look at it yourself on your textbook, page 935. So I'm going to briefly go over the chemistry of vision. All right. So your mom always says to eat carrots, and the reason for that is it's got tons of beta carotene, which is in your body broken up into vitamin A, which is also converted to 11-cis retinal. So this is what is used in your body for you to see. All right. So this process, beta carotene to 11-cis retinal, happens in liver, and it gets converted. So once you have 11-cis retinal, it reacts with an amine functional group. So this primary, as you can see, is a primary amine. Opsin is the protein, and off of protein is this amino acid that's got a primary amine attached to it, which we'll learn about this type in last unit. So it forms a imine bond. All right. Now this imine bond is sort of held together with this protein, and that is called rhodopsin. So rhodopsin then is right here, and because 11-cis retinal at this juncture, this carbon is a cis, not trans. When the light hits this double bond, notice all these double bonds are more transoid, right? But when the light hits this 11-cis retinal, it turns a cis into trans, as you can see over here, right? What that does is, I have a picture of this here. So this, think of this as the big protein, and this is the 11-cis retinal, and then the light hits. I'm gonna have to hurry up. So when the light hits, the 11-cis retinal, which is more like a cis here, like this, so it's sort of like a U shape. But then when the light hits, it becomes trans, so it kind of juts out. So this going from cis to trans causes this shape to change. All right, and when that happens, there's a nerve impulse that's sent to the brain saying that is perceived as a vision. All right, so because now it's no longer kind of protected, if you will, in the protein, because it's jutting out, it gets hydrolyzed easily, and it goes back to the opsin protein, and then it's ready to do the reaction again of forming the imine, and the light hits again, blah blah blah. All right, so that's how the impulse that's sent to the brain when the cis becomes trans is perceived as vision in our body.